All right. So for today's lecture, we're going to be building off of what was covered in the pre-lecture and trying to continue reviewing some of the basic cell structures within a given cell. We'll be looking at different structures, particularly that are a part of the cell membrane, what it's composed of, and how those different parts imbue certain functions to the cell membrane. And we'll be talking about all of this in the context of lupus, and we'll be talking about it particularly within the context of the pathogenesis of lupus. Pathogenesis typically refers to how the pathology of a given disease arises, how it begins. So the proper medical term for lupus is systemic lupus erythematosus. You might see it written as SLE, or just sometimes commonly referred to as just lupus. It is an autoimmune disease that's associated with antibodies against nuclear proteins. Does anybody want to take a moment to define what an autoimmune disease is for us? Yeah. Yeah, exactly right. It's where the immune system attacks its own body. Typically, our immune system is designed to distinguish between self versus foreign. Another way to describe autoimmune disease is where the self is identified as foreign and typically attacked. What is identified as the part of self that's identified as foreign is going to be unique to each autoimmune disease. And in the case of lupus, we actually raise autoantibodies. Autoantibodies are the antibodies in the autoimmune disease. We develop antibodies against our own nuclear proteins. Some of the symptoms associated with lupus are can seem fairly, I don't want to say benign, but rather can seem a bit vague, and there's going to be a reason why. So some of the symptoms involve fatigue and joint pain. There's oftentimes a degree of arthritis that emerges, that presents with some lupus patients. There's also a degree of photosensitivity among them. There's also this malar rash that occurs on the face of lupus patients. There's a particular reason the malar rash is not specifically only unique to lupus. It actually occurs in another instance, and it is tied to the name. Does anyone know where the term lupus originates from, or what it comes from, or what it perhaps might be associated with? Yes. Wolf, that's correct. Um, Lupus, the disease, was given its name because historically a 13th century doctor identified that when patients present with a wolf bite, they get this malar rash. And so when lupus was first termed, it was because this malar rash was associated with a wolf bite. There's also a fair degree of anemia that patients present with. Again, that's the loss of red blood cells. Um, a lot of these symptoms. There's also a degree of let me see if we get there. There's also some instances of organ failure. There's also patients can present with lupus nephritis, where they have inflammation of their kidneys, and so their disease can progress to a point where they go into kidney failure. But there's lots of diseases that can present with some of these symptoms, and that's why lupus is sometimes categorized as the great imitator, because it can be hard to diagnose until these autoantibodies, the antibodies against nuclear proteins, are identified with a blood test. Lupus alternates back and forth between flare-ups and periods of remission. When a disease is in remission, that's typically when there's no presentation of symptoms. And so patients will go back and forth between having no symptoms when they're in remission versus flare-ups, where they might be in a lupus crisis and have either really severe arthritis, have severe joint pain, present with some symptoms of kidney issues or kidney failure, and have nephritis. So the prevalence and incidence. It's estimated that the prevalence right now within the US is about 1.5 million people and 5 million people worldwide. Because lupus is the great imitator, because it can be hard to diagnose with some blood tests, without blood tests, I should say, sorry, forgive me. Because it can be hard to diagnose without a blood test, it's estimated that these numbers are an under projection or an underrepresentation of how many people currently actually have lupus within the US and in particularly worldwide. Um, It's also been noted that women are 9 times more likely than men to develop lupus. This is largely true for most autoimmune diseases that women have there's a higher prevalence and incidence rate among women for most autoimmune diseases. Why that's the case, it's not entirely clear. There's a lot of work in research going on right now to appreciate sex differences in biology and in particularly in disease and trying to understand the mechanisms behind them. Uh, and it's also noteworthy that non-Caucasian women or women of color are 2 to 3 times more likely to have lupus than Caucasian women. And so there's a bit of uh, a, a bit of certain communities being predisposed to having either lupus or, again, so this is similar to most autoimmune diseases. When we talk about the economics of lupus, the cost of the direct expenditures for lupus in the US is about $18 billion. Yes. Yeah, that's a great question. We don't know. There are certain genetic factors that that explain lupus. The environmental factors that are at play, we don't quite know yet. We're going to talk today about some of those genetic factors that that seem to predispose one to lupus, and perhaps one possible explanation as to why it's enriched within the United States, why it's overrepresented within this cohort, is that typically in communities and populations where these genetic alterations emerge, uh, that genetic pool it maintains that mutation and it doesn't typically go over and out into other genetic pools or other populations. That's one explanation, but at the end of the day, I, I'm not 100% sure why, but that's just a theory that could explain why that's the case. Um, so the direct expenditures for lupus in the United States is $18 billion. When we talk about direct expenditures, that's usually associated with treatment and therapy. Um, Indirect expenditures are usually secondary effects of the disease. So, for example, an example of a secondary of an indirect expenditure would be loss of income, could be trying to think of another example. Loss of property. It's just a lot of other secondary effects that are not directly tied to the treatment or therapy of the disease. The etiology, again, that's cause. Oh, yes. Mhm. Are you expected to memorize that for the exam? I would say it is helpful to, it would be good for you to know that there are populations that are enriched within lupus among lupus patients. Um, I'm going to try and make sure that I focus on the key information. So actually, this is a pretty good opportunity to maybe highlight something that was articulated in the introductory lecture, but it will be worthwhile to take 30 seconds to explain here. Um, this class, when you first start out, it feels like you're drinking from a fire hose. I've been in your seat. It is a lot. I'm very much mindful of the pace at which I'm going, as well as how much information I'm sending out to you guys. Um, the helpful way to think about this material is not to try and jot down every single word I say or every single piece of information, but rather try and think about what is the key information from each given slide, what's the take-home message, and then how do all of these things relate together and come together to form this coherent picture of uh, lupus and cell membranes and how they relate together, of amino acids and sickle cell disease and how they relate together. And that'll be helpful as we move throughout the course, because everything we learn in this first quarter, we're going to build on that and apply that to immunology. We're going to build on that and apply that to DNA replication and gene transcription. We're going to apply that to metabolism, glycolysis, oxidative phosphorylation. So I'm not suggesting that you sit and try and memorize every word I say in each recording or every single word that's written on each slide, but rather what is the key take-home messages, and how do they piece together to form a coherent picture of how these processes work, how they go wrong in disease, how they're treated clinically, and then even building upon that further in a research setting, how if we manipulate this, how might things change? How can this information that we're taking here relate to the previous lecture? Someone asked earlier about hydrophobic proteins and situations where they might not be, where they might be in a non-aqueous solution. We're going to talk about that today. So we're going to build upon what we learned last class and see a situation like that question where we might see a non-aqueous environment or a non-hydrophobic environment that a protein might be in. And so again, you're drinking from a fire hose. I appreciate it. I want to direct you to think about the key take-home information from each slide and how these pieces fit together. I'm going to try and do my best job to articulate what is the take-home message from a given slide, but you're also welcome to sometimes ask me, what's the take-home message here? I would very much appreciate that because sometimes I can meander on and go down a rabbit hole and could sometimes use an imaginary cane to kind of pull me back into focus. So with that being said, I'll jump back in. So we talked about etiology last class. That means the cause of the disease. The cause of lupus is largely unknown. This goes back to a question a student had a moment ago. There are certainly genetic factors that are at play with lupus. There are also environmental factors. What we so far know, and I should take a moment to say, when we have an uncertain etiology, the clinical term for that is idiopathic. That is a large fancy medical term for we do not know the cause. There's no known etiology for this. What we so far know about lupus is that the environmental factors at play do not cause the disease. However, they can exacerbate it and contribute to flare-ups. And so today we're going to talk about one potential mechanism that can explain how lupus emerges. But again, it's not a cold, hard, or it's not a cut and clear pathogenesis, but rather just one potential mechanism that can explain how lupus arises. Before we jump into switch gears into the content, are there any questions? All right. So today, a lot of our class is going to be focusing on primarily the cell membrane. We're going to be referring to a couple times, maybe perhaps a few other organelles, but we're going to be, the meat of the lecture is really focused on the cell membrane here. Some of this is going to be a little bit of a recap from the pre-lecture, but it really is worth taking a moment to explain a few things here. And so when we talk about the cell membrane, there are a couple key components here. We first have the lipid bilayer. The lipid bilayer is composed of phospholipids, primarily oriented this way, where we'll have the intracellular compartment and the extracellular compartment. So this is the inside of the cell and the outside of the cell. And these phospholipids are oriented this way. And we're going to explain in a moment how and why they're oriented this way. But we have phospholipids that are part of the lipid bilayer. There are also going to be cholesterol that's found within the lipid bilayer that plays an important part within the cell membrane here. A key thing to note is that the lipid bilayer provides the key functions that the lipid bilayer provides is it provides a physical barrier between the extracellular and intracellular compartments and helps to maintain concentration gradients. I suspect for those of you that are taking Physiology 1, you've talked a lot about and learned a lot about the specific concentrations of certain ions intracellularly and extracellularly. And the lipid bilayer, the cell membrane, helps maintain those gradients. The other very important key component of the cell membrane are proteins. Every other function associated with the cell membrane that's not barrier or not concentration gradients is provided by proteins. Proteins play an incredibly important role in functions at the cell membrane, and they provide cell-specific functions from different cell populations to different cell populations. What's really, really important to note here is that it's easy to try and use cell membrane and lipid bilayer interchangeably. You want to avoid doing that. The lipid bilayer is a component of the cell membrane, but they cannot be used interchangeably because The other key components that make up the cell membrane are molecules like cholesterol and proteins. So it's important to keep that in mind. Um, when we talk about the lipid bilayer and the phospholipids, oh, yes. Great question. You're asking good questions there. We're gonna take a moment in about 5 to 6 slides, I think, to describe how this phospholipid bilayer, how this membrane is present on other organelles within the cell. So hold that thought. Um, so these phospholipids that make up the lipid bilayer have a few key components here. So we have a fatty acid chain that's a long hydrocarbon molecule, and it's incredibly hydrophobic. So this is a hydrophobic tail that's called a fatty acid chain. We have a glycerol molecule and a phosphate group here. Um, these three pieces, the fatty acid chain, glycerol, and phosphate, are ubiquitous across all phospholipids. And we call those three pieces together phosphatidic acid. So kind of similar to our previous lecture on amino acids, there are features of amino acids that are common to all. There's the amine, there's the alpha carbon with a side group, and there's a carboxyl group on the end. So there's kind of a similarity or a resemblance there. What makes each phospholipid specific is the alcohol group. You can think of it similarly to the side group with amino acids. In this case, we have the alcohol group is a choline. So we refer to this phospholipid as phosphatidylcholine. Each phospholipid will typically begin with the name phosphatidyl and then the name of the alcohol that's specific to that particular phospholipid. So we have four alcohol groups that are typically present on phospholipids in the cell within the phospholipid bilayer. We have choline, serine, ethanolamine, and inositol. So you'd refer to these as phosphatidylcholine, phosphatidylserine, phosphatidyl-ethanolamine, and phosphatidyl-inositol. And so a key take-home piece of information here is that all phospholipids are made up of phosphatidic acid, a very hydrophobic fatty acid chain, a glycerol group, and a phosphate group. The last piece is the alcohol that is specific to each phospholipid. So if we had a beaker full of phospholipids within an aqueous solution, what would end up happening is that because of these long hydrophobic fatty acid chains, fatty acid tails, it would create a phospholipid bilayer where these fatty acid chains would orient together, and we'd have the polar heads here that also constitute of the phosphate groups, the polar heads on the outside and the hydrophobic tails on the inside. And so that's how and why our cell membranes with the phospholipid bilayer are composed of this way, where the hydrophobic regions are turned towards each other and the polar head groups are oriented out towards the extracellular region or outward towards the intracellular region. What's important to note is that this spontaneous formation of the bilayer is driven by these hydrophobic interactions. And what's also important to note is that if you look here, it's not a completely the way I've illustrated it. This is kind of the shorthand for how lipid bilayers are drawn, and these fatty acid chains are drawn as a straight line. In biology, those fatty acid chains are typically not straight lines. If you look here, there's these kinks or bends in some of the fatty acid chains, and that's incredibly important. And we're going to talk about why in a second. But before I move on, I'm going to stop for a couple of questions here. Yes. Does each phospholipid have its own alcohol group? So all phospholipids, so looking here, every single one of these phospholipids has the fatty acid chain, has the glycerol group, and has the phosphate group. But for example, I'm going to take a moment and confess I'm colorblind. So if I say colors wrong, forgive me. But for example, the specific colors here on the phosphate heads here, the blue and yellow, and I'm only seeing two colors, so forgive me if there's more than that. But you can envision that. Let's not do this. Um, I see red, but I'm not going to jump there. But you can envision those being specific to each species of phospholipid. So, for example, we could say the blue here is phosphatidylcholine, the yellow here is, for example, phosphatidylserine. And so you'll have multiple of those present throughout the phospholipid bilayer. But what makes each phospholipid specific is the alcohol group present on it. Does that help? Can't believe you all laugh like that in my colorblindness. Yeah, any other questions? All right. So when we talk about the cell membrane and cells in general, it's really helpful to talk about the scale of these things. It's biology happens in 3D space, so it's helpful to think about these things and know their scale, so you can sometimes envision or picture these things happening. So when we talk about the cell membrane is only about 4 nanometers thick. 1 nanometer is 10 to the negative 9 meters. Proteins themselves can range from either 1 to 100 nanometers. They can be really small single peptide chains. They can be large multi-protein complexes that are as large as 100 nanometers, but it usually is dependent on their mass and their shape. And finally, a typical cell, that's in quotations here because the size of a cell can be different based on what kind of cell it is. But on average, a typical cell is usually 10 microns or 10 micrometers in diameter. To help kind of give some context there, an average human hair, depending on how fine it is, but we could say that the average adult human hair is about 100 microns in diameter. So that would be about 10 cells. The human hair is about 10 cells thick in diameter. And so it can sometimes, again, be really helpful to try and envision this in space and see how these things relate and fit with each other. So this is another helpful illustration and snapshot of what the phospholipid bilayer looks like. We have, again, these polar head groups on top, say, this extracellular space, and the polar head groups down here in the intracellular space. They're polar, they have a charge. These fatty acid chains are hydrophilic, don't have a charge, so they orient together. And again, while the hand-drawn illustrations usually show straight lines, you can appreciate here at the kinks that are present within the fatty acid chain on these phospholipids. This is not a stationary structure. What I mean by that is that there's usually lots of wiggling and moving within the phospholipid bilayer. That is also temperature dependent. And so in a research lab, sometimes if we want to study what's happening on the cell membrane, if there's a certain protein or molecule on the cell membrane we want to study, we will put the cells on ice so that there's less movement, there's less movement occurring because proteins, and I'll show you in a moment what this looks like, that are wedged within or on top of the cell membrane can flow freely within the cell membrane here. That fluidity has been given a name. We refer to it as the fluid mosaic model. Yes. Hold that thought because that is a key piece of information we're going to talk about at the end. Great question. We're going to come back to that. The model that helps to explain this fluidity, this movement within the cell membrane is called the fluid mosaic model. So we have our proteins that are present within the cell membrane. They're embedded in this semi-fluid lipid bilayer. So the lipid bilayer is the fluid part of the fluid mosaic model. And these proteins that are sprinkled throughout the cell membrane are the mosaic part of this fluid mosaic model. And so what this looks like is that we have peripheral membrane proteins. There are proteins that sit on top of the lipid bilayer. So if we think about the fact that it's sitting on top of these polar head groups of a phospholipid, what kind of interactions might what's a good way to say this? What kind of interactions might be driving um, this protein interacting with the polar head groups here? Yeah, yeah, electrostatic or ionic interactions driving how this um, peripheral membrane protein sits on top of the polar head group here. Another class of membrane-associated proteins, bless you, is lipid-anchored proteins. Um, Despite the illustration showing that it kind of sits on top of the polar head group, a lipid-anchored protein has a lipid molecule associated with it, and that actually wedges down into the hydrophobic region of the phospholipid bilayer. Um, so what's key to note here is that the very middle here would be considered the most hydrophobic region of the phospholipid bilayer. So this lipid anchor would sit right down within in the middle of the phospholipid bilayer and situate that protein right there at the cell membrane. Another kind of membrane-associated protein is referred to as an integral membrane protein. These are proteins that are actually shoved into the cell membrane, shoved into the phospholipid bilayer itself. So seeing for a moment that, and this goes to a question someone had at the very beginning of the lecture, seeing that this protein is wedged within the internal part of the very middle of the phospholipid bilayer, what could we say perhaps about the amino acids that are probably present in this region here? Yes, exactly right. Those amino acids there on that protein would be hydrophobic. And so there are some integral proteins that actually traverse the entire distance of the cell membrane. And so these would have both, kind of similar to the integral, these would have both large hydrophobic regions and charged hydrophilic regions of this protein here. And as I described earlier, these proteins are not, this is not a stationary, um, this is not a stationary structure. So what ends up happening is that these proteins typically will not move in and out of the phospholipid bilayer that way, but rather they'll move in this direction around on a cell membrane. So if you think for a moment of a cell as a spherical 3D object, and if there's one specific protein that we're considering, this protein will sometimes just wiggle around on the surface. If it's a cell that's been put on ice, that protein will start to wiggle slower and slower and come to a halt. But these proteins will typically be moving throughout this semi-fluid phospholipid bilayer. They'll diffuse laterally, but not in and out like in the Z of the phospholipid bilayer. When we take kind of taking some things that we talked about last class and bringing it here, we talked about alpha helices, the secondary structure, and how there's two key features of alpha helices. There's hydrogen bonding that occurs vertically, and also these side groups are oriented all outwards. For integral transmembrane proteins, the those those proteins are typically comprised of alpha helices, the amino acids in the region spanning across the cell membrane are going to be alpha helices where their side groups are sticking outward. And because they're within the middle of the phospholipid bilayer, they're going to be hydrophobic. And so these integral transmembrane proteins are going to have hydrophilic alpha helices that are present within the hydrophobic region of the cell membrane. What's important to note is that as these hydrophobic non-polar side groups are facing outwards, the polar backbone is going to be buried internally, protected from the hydrophobic region there. So if we take a moment to consider this, um, we know that I'm going to write some of this information on the board. Given that we know that the diameter of the thickness of the cell membrane is 4 nanometers, and given that we have for each turn, that's about half a nanometer, and for each turn we have 3.6 residues or amino acids. For a transmembrane protein, it would require a stretch of how many hydrophobic amino acids across the cell membrane. So we're going to do a quick exercise to do a quick back of the napkin calculation. So go ahead and try and figure out, knowing that the thickness of the cell membrane is 4 nanometers, written on both sides here, and given this information that for each turn each turn is half a nanometer, and we have 3.6 amino acids for each turn, how many amino acids do you think would be present within that region, or how many is required to stretch across the cell membrane of for a transmembrane protein? So do a quick back of the napkin calculation. Or saying like for a quiz, an exam, that information would be given to you. So we'd say, um, given a given a 4 nanometer diameter or thickness of the cell membrane, then this question would be asked. Does that make sense? Yeah. All right. Does anyone want to share their answer here and how they got there? Yeah.
I got around 25. So each turn there's 4 nanometers of space. So essentially, with 0.5 nanometers per turn, there's 8 turns. So just multiply 8 by 3.6.
Yeah, that's exactly right. So for each transmembrane protein, there would need to be about 25 consecutive hydrophobic amino acids. So when we think about the primary structure of a protein, the written out amino acid sequence, could we potentially identify membrane spanning regions of a protein just purely based on its primary structure? The answer is yes. There's actually a tool that helps us do so. It's called a hydropathy plot. And so what it does is it looks at specifically the primary structure of a given protein, and it's plotting the hydrophobicity, whether it's more hydrophobic or less hydrophobic, where that red box is this sweeping box. The red is at the single amino acid, but these gray sides are also considering the hydrophobicity of adjacent amino acids. So if we're starting at the N terminus of a given protein, as it goes, as we see it being more hydrophobic throughout the course of the amino acid sequence, it's going up. And then it comes back down and becomes less hydrophobic. So if you were to imagine, I can't reach over there, but if you were to imagine a protein that's being read at the primary sequence level, the less hydrophobic regions could be either the extracellular or intracellular. Sorry, I should say the peaks in the less hydrophobic region could conceivably be either extracellular or intracellular regions. But the key thing to note is that for each peak that goes up and comes back down, it's important to note where do we think the top of the most hydrophobic peak is physically located in the phospholipid bilayer. Does anyone want to venture a guess where the most physically where these locations would be within the phospholipid bilayer, the top of those peaks? Yes. In the bilayer. Would you say, I guess like option one could be like here, here, or here, like the top of the very peak. Where do you think that? Yeah, it'd be right in the middle. So the peak of the more hydrophobic regions corresponds to the very middle of the phospholipid bilayer. So as we're looking at this peak, we could think of each complete bell shape here in the hydrophobic region being an entire uh, membrane spanning region of a protein. So as it's sweeping across, say we start in this extracellular region, as we go through across that one peak, we're going down into the cell, or into the cell membrane, forgive me. And then as it gets down here, we're in a more, forgive me, as it gets down to this bottom peak in the less hydrophobic region, we are now outside the phospholipid bilayer by the polar head groups. We see that it's going back up to another peak in the more hydrophobic region. So that would be actually coming back up here. And so taking a moment to see the number of peaks in the more hydrophobic region, how many times do you think this specific protein spans across the cell membrane? Four is correct. So there are four membrane spanning regions. Again, a shortcut would be to think about the peak here corresponding to the very middle of the phospholipid bilayer. So again, if we're starting going down into the phospholipid bilayer, coming back out, this would be going into the very middle and coming back out. Going into the very middle, coming back out. That's two spans. Going into the very middle, coming back out. That's spans going into the very middle and coming back out. So that's four times that this specific protein, based just on the primary sequence, would span across, be predicted to span across the cell membrane. This specific protein is part of a class of proteins called tetraspanins. Tetra for spanning the cell membrane. And so we can use a hydropathy plot to predict what the physical structure might be of a given, what it might be of a given protein. Are there any questions here on this hydropathy plot? Yes. Yeah, so great question. So because this is in the less hydrophobic region now, this is going to be the C-terminal end of the protein that's down within the intracellular compartment of the cell. Great question. I should have clarified that. I appreciate you asking that. Yes. Yeah, the red box is at a given amino acid that's being read. So if actually, there's an animation, I believe, here. So it sweeps across, reading each amino acid at a time. The gray boxes are also weighing the adjacent amino acids and their degree of hydrophobicity, and predicting whether this region, when it's at the red box is at a certain position, how hydrophobic this area of the protein is. Does that help clarify? Yeah. Where would it be perfectly zero? Um, yeah, no, that's a great question. Um, Potentially, I think it's yeah, because you're getting to this charged polar head group here, as you're coming back out, peak of the more hydrophobic region. As you're getting towards the tail, this would be peak of the less hydrophobic. So I'd probably say between here and here, zero would probably be close to the base of the polar head group where the fatty acid chain meets. Um, but yeah, I'm gonna keep moving because I'm gonna try and cover everything. We'll see if we can get there. Um, so uh, the membrane function that's attributed to the phospholipid bilayer here, it's relies heavily upon the proteins associated with this with the phospholipid bilayer. So again, the phospholipid bilayer is a physical barrier and it maintains concentration gradients. all the other functions attributed to the cell membrane are driven by the proteins present in the cell membrane. Um, some of those functions, and this goes to another question we had earlier about other organelles or other membranes within the cell. There is a compartmentalization function that the cell membrane provides. Um, Part of that compartmentalization is a localization of function. An example of that is the nucleus. The nucleus there's a nuclear membrane, a phospholipid bilayer, and there is gene expression, there is transcription occurring within the nucleus. So we have compartmentalized function within the nucleus. Uh, we also have lysosomes that have a membrane where things are being hydrolytically degraded within this lysosome. We also have the raising of concentrations on or within a given membrane. And so an example of that, again, is the lysosome, where ion concentrations are being regulated by this membrane to mediate the degradation of what's within the lysosome. We're going to go into a lot greater detail of lysosomes later on in the lecture, later on in the class. But just for now, think of it as an organelle that where some degradation happens, and it has a membrane that mediates that function. We also have the regulation of transport. Some of the proteins that are on the surface of the cell membrane are actually shuttling molecules in and out of the cell at all times. And so that's mediated. These proteins help mediate that shuttling. We also have the binding of ligands and receptors. So we have receptors on our cell surface that are these proteins that bind to a specific molecule. That's called its ligand. Receptors bind to a ligand. And so that ability of a receptor that's a protein to bind to a ligand is incredibly important and can help transmit signals down into the cell that drive so many of the functions that our cells have. Part of this binding can also be between cells or a cell to a substrate. So our cells are usually in an environment called the extracellular matrix that's largely comprised of collagen, but other proteins. The ability of a cell to bind to its environment is mediated by receptors that bind to collagen or these other extracellular matrix proteins. So the degree of fluidity is really key for these phospholipids. If the membrane becomes, if the phospholipids within our cell membrane, within our phospholipid bilayer, if they became too quote unquote solid, that would jeopardize the function of the proteins in our cell and thus jeopardize the cell itself. And so it's really, really important that this fluidity be maintained. This is where that kinking that I referred to a few times of the fatty acid chain becomes really important. And that kinking is determined by the degree of unsaturation within that fatty acid chain. Does anyone want to help provide a definition of unsaturation or degree of unsaturation? Yeah. Yep, exactly right. It's the number of double bonds. So the more double bonds present on a fatty acid chain here, the more unsaturated it is. So for example, we have stearic acid here, which is animal fat. It has no double bonds present there. So it would be a totally saturated fatty acid. We have olive oil and we have aloic acid and alaitic acid, or olive oil and Crisco. Both have a double bond. And because of this double bond present here, there's a kink introduced. You might note that there is a double bond here, but there's no kink, and that's because of the nature of that double bond. The different double bonds that emerge can be either in cis or in trans. When we talk about cis and trans for these double bonds, that's meaning the arms of the double bond, what direction they're pointing in. So in aloic acid here, both arms of this cis double bond are pointing in the same direction, the same orientation. So that's called a cis double bond, and that introduces a kink. Whereas the trans double bond here, both arms are oriented in different directions, meaning that there's no kink introduced here. So the degree of unsaturation is not the only important thing, but rather the orientation of that double bond, whether it's in cis or trans, is of great importance to us. Because if we took a moment to think about if all of the phospholipids in our cell membrane and our phospholipid bilayer, if they were all comprised of stearic acid, that would potentially be a problem because the degree of order and crystallinity of that fatty acid would be such that there's no movement available at all. We want to decrease crystallinity and introduce less order and structure by having these kinks present. And so both stearic acid and oleic acid would not be ideal fatty acids present within our phospholipid bilayer because of the degree of crystallinity, the degree of order. We want there to be less crystallinity. We want there to be kinks that facilitates movement. If you think about animal fat and olive oil and how this crystallinity relates to movement, think about what happens when you put both of these items in a fridge. One solidifies and is oriented intact at a molecular level, packed tightly, whereas olive oil is still fluid. There's a lower degree of crystallinity that still enables movement, whereas animal fat, totally saturated, much higher degree of crystallinity, does not facilitate movement. And so that's going to be really key to think about the phospholipids that are present within our cell membrane. Cholesterol is an important factor in mediating some of this fluidity. So there are lots of different kinds of fatty acids that we can consider that have different degrees of crystallinity. So taking what we talked about a moment ago, of these pairings of not amino acids, of these pairings of fatty acid chains, which would we prefer to have within our phospholipid bilayer? Does anybody want to make a case? You want to go for it?
And on what environment you're in. In general, the second two, if
Mhm.
you want higher.
Yes. Well, if you think about it could depend on temperature, but I would pose the question in general of these two, which pairing would we prefer to have within our phospholipid bilayer? The bottom two. There's less crystallinity, there's more kinking that would facilitate more fluidity within our cell membrane. Whenever we talk about fatty acids or lipids and you hear omega fatty acids, that usually refers to the number carbon at which you find the first double bond. So omega-3 fatty acids have the double bond at the 1, 2, 3rd carbon, and for omega-6, it's going to be at the 6th carbon here. We're almost done. I'll get us there. We might go a couple minutes over, forgive me. Cholesterol plays an important part in maintaining fluidity. If we took a moment to imagine a phospholipid bilayer looking down upon it and envisioned this perfectly crystalline ordered structure at lower temperatures, there's going to be less movement, and there's going to be less, and there's going to be typically more crystallinity there. And so cholesterol being present within the phospholipid bilayer helps disrupt that structure and help provide more fluidity and less crystallinity there. In instances where there's potentially a much higher temperature, where there's going to be way more movement, we don't want there to be such movement or fluidity where these phospholipids are totally separated from each other, because that would disrupt the cell membrane, that would jeopardize the integrity of the cell membrane. So cholesterol, through the hydrophobic interactions with the fatty acid tail of these phospholipids, keeps these together. So at higher temperatures, it changes the crystallinity to make it less structured and ordered and increases the fluidity. This is at lower temperatures. To the far right, at higher temperatures, it maintains the integrity of the cell membrane through hydrophobic interactions with the fatty acid chain. There's regions within our cell membrane that are cholesterol rich, called lipid rafts. There are these small nanodomains that have a ton of cell signaling that are really important structures that we study in biology. And finally, taking this all back to lupus, there's one specific phospholipid of interest here. That's going to be phosphatidylserine. There was a question earlier about the flipping of these phospholipids. Certain phospholipids are predetermined to either just be on the outer leaflet or just be on the inner leaflet, or they can sometimes be on both. One that we're particularly interested in is phosphatidylserine. That is an inner leaflet phospholipid. There's a process by which our cells die. It's a programmed cell death. It doesn't happen by like mass tissue damage, called apoptosis, where what happens is there's the flipping out of phosphatidylserine from the inner leaflet to the outer leaflet of the phospholipid bilayer. That usually serves as a signal and cue to other cells that this cell is dying and we need to get rid of it. During this process of apoptosis, programmed cell death, there's also this blebbing, this taking of intracellular compartments of the cell and releasing it in blebs on the surface of the cell. What's important to note in what typically happens is that there are cells that eat other cells or other molecules called phagocytes. One example is a macrophage. So when we have a cell undergoing apoptosis, phosphatidylserine is flipped to the outside, and that is a signal for the macrophage to eat it and get rid of it. And it processes it and disposes of it. What typically happens in lupus is that there is a disruption in the ability of phagocytes to perceive this apoptotic cell needing to be disposed of. That's the first issue. So you can think of it as the garbage men going on strike. These phagocytes are not clearing out this apoptotic cell that has phosphatidylserine flipped to the outside, which is a cue that this cell needs to be disposed of. The other issue is that this blebbing, this release of nuclear proteins are now being identified. They should be identified as self, but they're also now being identified as foreign. So the two main issues is that there's not the disposing of apoptotic cells, and that the blebbed out nuclear proteins are now being identified as foreign. Those are the two key issues here at play with lupus. If autoimmunity or cell death really interests you, we have Kenneth Tung, who is a leader in the field. There's also a great professor at Penn, Cornelius, who is leading the field in studying cell death. If this interested you at all, I'd highly recommend you reach out to them. I'm going to hang back to answer any questions. Sorry for going 6 minutes over. That's on me.